Hello, everybody. My name is Martin. I just returned from the conference of animal behavior and ecology in Taipei, where I presented this talk. It was actually quite nicely accepted, and there were many questions from many people after the talk. And some people told me that they are a little bit unhappy that they missed the talk and asked me for presentation. So I decided to record it. I changed few slides a little bit based on the questions I got, so this is kind of a nicer updated version actually. Okay, so let's go. I will remove my face. So the presentation is about the topology-constrained phylogenetic analysis. And we were trying to do it on two Triassic fossils, these two beetles. Both studies are already published. One is in Systematic Entomology 2019. One is in Current Biology 2021. The Current Biology was quite popularized or visible in the public because it is a beetle in the dinosaur coprolite. However, in both cases, the important thing for me was that these beetles are Triassic. Triassic is a very important period for beetle evolution because many modern lineages started to diversify or appear at that time. But unfortunately, we don't have many fossils from the Triassic. So every fossil is actually very important. It can tell us a lot about evolution of beetles, and that's why we have to try our best to figure out where they actually belong to, what is their phylogenetic position in the beetle tree of life. So why we actually need it? Why we need to know the phylogenetic position of the fossil. Okay, because we are curious and we want to understand the diversity of beetles in the past, but in reality, the direct usage of the fossils very often is that they are used for calibrations for dated phylogenies. Uh, because we need actually dated phylogenies to study any aspects of evolution. So you can find studies which are totally far from any fossil things, like this study on genome evolutions of beetles which went underground. This is nothing in common with fossils. They are studying the changes of genomes in these beetles using some very cool modern sequencing methods and bioinformatic tools. But as you can see, they also need dated phylogenetic tree because they need to put their results, put their data about genome changes in the context of time. And that can be only done if they have the dated tree where the branch lengths actually correspond to the time which passed between the diverging events in the tree. So even in studies which have nothing in common with fossils, fossils are actually needed because fossils are most frequently used way how to get the dated molecular tree. There are actually two basic methods for how to do it, how to date a molecular tree. The kind of newer method is called tip dating, or sometimes also total evidence dating. The idea is that you actually mix together the modern species, which are the black ones in the matrix above, and the fossils, and you also combine the morphological data set, this is the 0, 1, 2, and the DNA data, and you actually run everything in one analysis, and you get the tree with the phylogenetic position of the fossils. You know their ages, and it also, based on these ages, makes or estimates the dates for the ancestor of the tree, so you get the dated phylogenetic tree. The second possibility, this is the kind of older method, is called no dating. So in this case, we actually make the phylogenetic tree of modern species only based on the DNA data, and we use the fossil to calibrate some of the nodes, some of the ancestors. Based on that, we estimate the ages of all the nodes, all the ancestors, and we produce the dated tree. The first method is very nice because except of the usual things which we can kind of include into the analysis like speciation and extinction, we can also include things like fossilization process or sampling strategy. There are special models developed for that, for example, the fossilized birded model, FBD. However, there are also some disadvantages. So one thing is that we actually need the morphology and DNA data for the same taxon or for the same taxa. This is not that frequently possible, not that frequently available. So this is the first obstacle. And the second one is that if you would like to have the tree corresponding to the real phylogeny or probably the most correct phylogeny in beetles, even in the deep divergences, even in the very early events in the beetle tree of life, then you would not be able to do it with few genes. You would actually need genomes. You would actually need thousands of genes. And that means that your data would be huge, your analysis will be slow, taking very long time, and you will also need lots of bioinformatics to prepare your data. So it can be actually not really the most practical way if you want to test the position of a single fossil as a paleontologist. So when we use the older method, the node dating, it is actually easier because we only use the modern species and the DNA. But we actually need to be very careful about this step when we kind of figure out which nodes are dated or calibrated by which fossils, because we naturally need a correct placement for the fossils. Otherwise, you can make a mistake that you use the fossil to calibrate incorrect node, and naturally, then you can get quite a biased time tree. So ideally, for this step, we would actually need some way which can test the phylogenetic position of the fossils which we are using for calibration in node dating. This is naturally not that easy. One of the problem is that the fossils are always fragmentary, so we never can actually see all the characters which we can see in the modern beetle, because some of them are simply not visible, some of them are hidden inside of the body, some of them are not accessible, some body parts may be missing, and so on. So we actually don't have all the characters, and that makes the phylogenetic analysis a little bit tricky. So unfortunately, at least in beetle studies, very frequently the phylogenetic placement is not tested at all. As an example, I can show you this study. This is a description of a new family of Cretaceous beetles. You can find in this paper this phylogenetic tree, but you cannot find there any analysis. So this tree is basically just written by hand. It is more like a hypothesis done by the author, but it's not not based on any real analysis. This is not, unfortunately, a very frequent way how kind of phylogenetic treatment is still done in many insect fossil papers. However, more frequently people try something which is cool. They code some morphological characters. So this is an example of another new beetle family described by the same group actually. And somewhere in their supplementary materials, you can find this small tree based on several morphological characters analyzed by maximum parsimony. You can see that the new family which they are describing is resolved as sister to something called Trochidae, but actually they don't trust the results at all. So if you actually check the paper, you can read that we know that the results of our morphological phylogenetic analysis must be interpreted with caution, since most deep relationships within the tree are incongruent with recent phylogenomic data. So basically, it means that the relationship between the clades they got is incongruent with what genomics showed, and that's why they don't really trust that the new family sits where their maximum parsimony analysis has shown, and as a result, they make another tree. So in the paper directly, you found the tree, the colorful one, and as you can see, this is again just handmade tree where the new family is sitting in a totally different position than resolved by the real analysis. So naturally, this is the way you can see that you can still publish this way in a very good journals, but it's naturally not optimal from the scientific point of view. So we ideally need some analysis which actually can go with morphological data only, because fossils actually have no DNA data. At the same time, it would be cool if the analysis can reflect the results of DNA-based or genome-based analysis. So we somehow can put in the results of the big data analysis which are available. Naturally, this is uh, not very practical to do it directly, because you will need lots of editing, processing, bioinformatics. It will be very time consuming if you do it directly in your analysis. So we have to find some way in which we can include this information without making our analysis too complicated. And ideally, we will be also happy if the analysis can show us something about the uncertainty of the phylogenetic placement, because if we have the fossils in which we cannot see all the characters, naturally sometimes we are not totally sure. There can be more alternative placements based on the characters we can see. So the usual result that the analysis just kind of runs and shows a single position is actually, in this case, in the case of fossils, not the optimal one. We would optimally like to get several alternatives which you can discuss or which you can further explore. Naturally, in case that your data are not strongly supporting a single position only. So that's why we were kind of trying to figure out what to do, and we started something which we call constraint topology approach. The idea of that is that first you screen the phylogenetic trees which are published about the group you study. So in our case, it was beetles. So we actually screened several genomic papers with the beetle phylogenies at the family levels. There were some updates. Some people sequenced additional species and added them in the previous trees. Then for several families, we have the internal phylogenies based on smaller gene selection. And in some cases, I think we actually, for the relationship inside of the families, even use the results of the morphology analysis, because there was nothing else available for these families. Nobody yet studied the internal topology of some families using DNA data. So we actually combined many papers and created something like guiding tree, created a tree which showed the most up-to-date idea about the relationship between modern species, and used this tree as the guiding tree. So this tree was fixed. The topology of this tree was fixed. Then we used a big dataset, the morphological dataset from John Lawrence et al., published in 2011, and we coded the fossil in. You can see that the fossil has many question marks, so these are these characters which cannot be coded. And then we run the analysis in which the fossil is the only species, the only taxon, which is allowed to move across the tree freely and sit anywhere where the morphological data support it. So you can do this actually in two ways. You can do it in the Bayesian analysis, and you can do it in the maximum parsimony way. So for the maximum parsimony way, I did it in TNT. It's actually quite very easy. You need the reference tree and the morphology matrix. Naturally, as in most maximum parsimony analysis, you get just single most parsimonious position of the fossil, which may not be totally realistic in case of fragmentary fossils. And moreover, it is a little bit difficult to measure how much you can trust the result, because the usual way we use for maximum parsimony is bootstrapping. But if you have lots of missing data, the bootstrapping actually is not very accurate way to measure that. So, but still, it's possible. For the Bayesian inference, I did it in Mr. Bayes. You also need a reference tree, which which has to be translated to the set of topology constraints using R script. And then you need a morphology matrix, the same as in the previous case in maximum parsimony. And naturally, normally in Bayesian inference, you also get a single final tree. This is so-called maximum credibility tree. And in this tree, you can get a single position of the fossil, which is the most probable position of the fossil. But actually, you can also explore the trees and check the alternative positions in which the fossils was revealed, and also get the posterior probabilities for it. So as you can see on the picture, which is kind of a schematic illustration, in this case you can see also this fossil was in 70% of cases revealed as sister to Aphodeus, in 20% of cases to Meloa, and 10% of cases to Tenebrio. So it can give you some alternatives, including the probability of these alternatives. How we get it? So maybe you know that in Bayesian inference, you run the analysis and you always get this trace log of the log likelihoods of some parameter values. And you know that we usually trash the starting phase as a so-called burn-in. And in the typical Bayesian analysis, you then take all the trees which were kind of explored during the MCMC search through all these hundreds or thousands or millions of generations for which you run the analysis, and you summarize all these trees into one. So this is the maximum credibility tree. But what you can also do is actually explore in this individual trees which were kind of searched during the MCMC run, and check what are the alternative positions of the fossil in this tree. And this can be actually mapped on the tree using the R script. So it's not that difficult, even though you have to kind of explore thousands of trees. So what you can get, here are some examples of some analysis we were trying when we were kind of testing this method. So naturally, if your fossil is very nicely preserved and the characters you can code are nicely informative and not in conflict, then you can get single highly probable placement. You can see here the red branch goes to some quite deeply nested node. So you can actually, with very high probability, say, okay, so this fossil probably belongs to this small clay. The colors mean the posterior probability, so you can see that the red branch is something like about 0.9, around 90%. More frequently, however, you get something like the middle picture, few moderately probable placements. So in this case, we got two alternative placements, each of them with moderate support. And naturally, in this case, it seems to be not the optimal way, but you can discuss and compare and further explore whether you can confirm or reject some of these two possibilities. So anyway, you still reject so many possible placements in the tree. So all of the out of the all the branches which the huge tree has, you actually now only have two alternative positions which you can compare, which naturally makes the work still much easier than to just compare without any guidance. The third possibility is that you get many weakly supported clades. This is the right tree. This naturally is not the optimal result, and it basically shows that your fossil does not have enough phylogenetically informative characters. So basically, you cannot say much about the phylogenetic position of this fossil. So all these things about the methods are summarized in this paper. You can go there, go to the supplementary materials if you want to have the guidelines or some kind of step-by-step tutorials for how to do the analysis, both the maximum parsimony and the Bayesian one. Or you can actually message me at any moment. I can help you to try if you want to try. So now I will show you just some examples which we analyzed using these methods. So the first one is Lee-Hermania, so it's a late Triassic fossil from USA, from North Carolina, I think, from Cow Branch Formation. And for a long time, it was considered as the oldest rove beetle, and it was included or used as such, even as a calibrating fossils for some very famous time trees. For example, even for the time tree of the complete insects, the first genomic study of insects by Misov et al. 2014. However, for a long time, some people were suspicious that this may not be rove beetle at all. So our task was actually to try to analyze the position of the fossil and get some more clear uh, solution of what it can be. So we were trying to code the morphological characters. After a very detailed and long analysis of all the fossils which we saw, we were able to code 95 of 344 adult characters in this John Lawrence morphological dataset, so about 27% of the characters. And when we run the topology constraint uh, analysis, we actually, of these three possibilities, which I have shown you before, we got the middle one. We got these two alternative placements. One was in the Staphylinidae, which is the rove beetle, so it basically corresponded to the original idea of what this beetle is. But there was an alternative position, which was the small group of beetles called Mixophaga. And the idea was, or the analysis has shown that it's most probably somewhere close to the modern family called Hydroscaphidae. So what we actually did, that we used the result of this analysis and we further explored which of these two placements can be more probable. So we did lots of detailed analysis of morphological characters. We are trying to search something which supports or rejects one or the other position. And we, for example, found this. So here in these fossils, this structure marked in red was considered as mandible by the original description, but we actually found out that it has segments. So that's not possible. In insect, mandibles are always one piece, not segmented. So actually, these things are maxillary pulps, not mandibles, and we have shown that mandibles are actually not visible. They are kind of hidden inside of the head capsule. This is not congruent with rove beetles. Rove beetles have always visible mandible from outside, but it's congruent with this Mixophaga beetles. So it was the first clue that actually the Mixophaga alternative can be correct. And then we did naturally lots of other comparisons of antenna, of genitalia. I did thousands of slides of both Mixophaga and the Staphylinids, also some scanning electron microscope things, and we found enough evidence saying that or showing that actually this beetle is really a member of the group called Mixophaga, and most probably it is quite related to this beetle on the right called Hydroscapha. So we put it in the same family at that time. The second example is the beetle from the dinosaur coprolite. So this is from the Polish locality Krasiejow. My Polish colleagues got a very cool idea that they were actually scanning the coprolites of these dinosaurs using the synchrotron, the micro CT with very high resolution. And they found lots of remains inside, including these cool tiny beetles. And my task was to figure out what this beetle is. Naturally, the most cool thing and the reason why it went to current biology was that they actually realized and discovered that coprolites can be used in the similar way as amber. So they can be also a very cool source of three-dimensionally preserved insect fossils. The only problem naturally is that they are not transparent, so you have to do the micro CT scanning and kind of extract the fossils out of them in the digital way. So they did very nice kind of work with scanning and extracting the data, but yet even through the work was very cool, the beetle was challenged. So you can see that even through the reconstruction is very detailed for the beetle, which has about 1 millimeter. It still is kind of limited in resolution for some very detailed tiny characters. So it was a little bit tricky. And we also had many disarticulated beetle parts uh, of the same beetles in the same coprolite. So we were at the end able to code 77 of 344 characters, about 22%. And we tried the analysis again. So in the first analysis, which was run without any additional kind of limits, we got four alternative placements, all of them in the huge lineage of beetles called Polyphaga, which actually comprises most beetles you can find outside of here nowadays. The problem is that when we were superimposing the disarticulated pieces of these beetles, we realized that this beetle had to have the blue part exposed. So there's this blue part on the prothorax, which is called propleuron. This had to be a separate scleroid, which was exposed externally. And as you can see on the drawings for Polyphaga, this blue part is never visible. So actually this character somehow falsified and rejected all four alternative positions which we got in this first analysis. So what I did in the second step, I actually bent the Polyphaga. So actually kind of constrained Polyphaga to be monophyletic. That means that the fossil cannot go inside during the search. And in this case, you can see that we got one very strongly supported placement, again in the beetle lineage called Myxophaga, and again close to the beetle called Hydroscapha. We then actually used another data set to explore more in detail the phylogenetic position of the fossil within this Myxophaga. The reason is that this big data, this big tree, are characters for family level relationships, and there are fortunately, there are some morphological data sets directly for Myxophaga with many additional characters. We actually use them. And the conclusion was that we actually nowadays cannot really be sure what is the phylogenetic position of this beetle, especially because there's a huge conflict between the topology between morphological and genomic analysis, and we are still not sure what are actually even the relationships among the modern families. So that's why we are not actually able to say with certainty where this fossil uh, was situated in this Myxophagan tree. Yeah, so these are the two examples. And as you can remember, we used something like 27 or 22% of characters coded. So actually, when I was kind of testing this method, I also was concerned if it cannot be biased when we have not when we have so little portion of the characters coded. So I tested it. The way how I did it, I randomly selected 10 beetles of 10 modern beetles, which were coded in the tree and in the matrix. So these are modern beetles, so we know their phylogenetic position based on the genomics and morphology, and we have their full morphological data. And I randomly deleted large portion of characters for these beetles. So I did several analyses. I kept 30%, 20%, 10%, and 5% of characters. And then I ran all the topology-constrained analysis again to compare where this beetle with deleted characters will be resolved and whether it will be actually close to the real correct position. Here is the result. So the blue line is the position revealed by the maximum parsimony. The red line is the position revealed in the maximum credibility tree in the Bayesian analysis. So this is the most probable placement base found in the Bayesian analysis. And the gray area is the area if we consider actually all the alternative positions revealed by this Bayesian analysis. If you go on the y-axis, the closer to zero you are, the more accurate you are, the more close you are to the correct position. And on the x-axis, you have different amount of coded characters. And you can see that when we remove more and more characters, then the accuracy drops down. But surprisingly, the drop down is only starting in some fossils around 20 or 10% of characters. So if we go under 20 or 10% of characters coded, under some I don't remember how many characters was it, under 60 or 30 characters coded, then it starts to be a little bit problematic. But then if you see the gray area, so these Bayesian inference considering all the alternatives, we can actually frequently get very close even with 10% data coded, so even with 30 characters coded in the matrix. So basically, it shows that the topology constraint methods seem to be robust, even with lots of missing data, because they only work with the morphology data, so they are quite easy and quick way to test the phylogenetic placement of the fossils. They allow to combine results of genomic analysis and morphological data, moreover, without the need to really work with the genomic data. And the cool thing is that the Bayesian approach can provide alternative placements, which can be further tested or discussed in your paper. Cool. So that's it. Thank you for your attention. And if you have any questions concerning this method, or if you need help with your analysis, you can always contact me by email. Thank you very much. Hope you enjoyed, and uh, good luck with your fossil research.